All right, this is the draw photosynthesis. This is we're going to tie this in with the draw respiration. I just did draw photosynthesis. What I want to show you though is this represents just a smaller section of the mitochondria. That's not very good arrows there, but you get the idea. So when we talk about photosynthesis, this is going to be our intermembrane space. Sorry about my handwriting. I don't have the nice fancy bamboo, which is the name of the program, as some others do. This right here is the cristae, which is named for the membrane. It's AE. So this part here, and this all in here would be the matrix. So again, all this represents this little section right here. All right. So we've got the intermembrane space. We've got the matrix, the cristae. I'm going to change the colors up here a little bit. So this part right here, we're going to call this pretty much right here, from here to here, is called the ETC, or electron transport chain. And it's basically what it does, it transports electrons. We've seen this before. This is our ATP synthase, and I may have spelled it wrong when we were doing photosynthesis, but we know it's an enzyme. There's the ACE. We can probably get the idea of what it does. I'm looking off of my sheet here, so if I'm looking off to the side. So it's electron transport chains, transporting electrons, but there's some things that are made here, and I'm going to have to get rid of some of this stuff. So hopefully I'm going to have to get rid of this. So we've got part of that labeled, but what I want to add in now is that we've got some carrier molecules that are important. I'm going to actually add 2 of them in here. So we've got NADH now, not NADPH, NADH and FADH2. What's going to be happening is, just like in photosynthesis, and this will be our ADP. Just like in photosynthesis, we're trying to produce ATP here. So we've got to create a concentration gradient. So we've got our membrane and all that. If you notice on these, we got NADH and FADH2, and hopefully you kind of see that they're carrying lots of hydrogens. If you remember this over here, the ADP and the ATP synthase, it produces ATP, but what has to go through it? The hydrogens. So the NADH and FADH2 are very important because these are what we're going to call, these are going to be carrier molecules, but they're going to be donating their hydrogens. So what's going to happen is, as the H pluses, these carrier molecules, they're donating these H pluses. So what's happening is, as electron transport chains occurring, these hydrogens are being forced across. So they're going across the cristae. And what's going to happen is NADH will become NAD, FADH2 will become FAD. I'm going to clear my space here. But we also have something here. I'm going to have to do this in a different color, because this is oxygen. There's some O2 there. But we also have hydrogens out here, by the way. So what happens is NADH will donate hydrogens, become NAD. FADH2 will donate hydrogens, become FAD. So now you've got a bunch of hydrogens up here. You're forming your concentration gradient. But one of the products of cellular respiration is water. Well, where does water come from? Well, we've got this O2 here. We've got some hydrogens here. O2 right there. So oxygen picks up hydrogens here to form water during this part of respiration that I'm showing you. Well, anyways, so hydrogens. Eventually, you're going to get to a point where you're going to have chemiosmosis occur again. Again, all these hydrogens, high concentration to low, so they're going to want to go through this ATP synthase again. Sorry about my handwriting. And as those hydrogens move through, ADP will become ATP. Big star there. Now, one glucose molecule, which we'll see here in a second, I'll do that here in a little bit, but this was kind of sloppy. So hopefully you get the idea that you've got this intermembrane space up here, you've got the cristae here, you've got the matrix here, NADH dropping off hydrogens, FADH2 dropping off hydrogens, oxygen's gonna come in, pick up some hydrogens, form water, form your concentration gradient, chemiosmosis occurs, and a bunch of ATP is made. And again, this is a simplified version. AP Bio, we go into a little more detail. I keep talking about AP Bio, but you've already hopefully seen this by the time you get there. Now let's scroll down a little bit. I may have to make this a little bit smaller so you guys can see everything. So I apologize. Let's see, maybe just have to cut it in half. Excuse me. All right, so now we're kind of showing you some other parts of the process. So over here is glucose. 1, 2, 3, 4, 5, 6 carbons. What's going to happen is this is essentially glycolysis. Glycolysis is the breakdown of glucose. We got 6 carbon. Gonna have to erase this. So what we have here, we've got ATP, 2 of them, which will form ADP plus a free phosphate. We've got ADP, which will form which will actually form 4 ATP. So as you can see, we're not really we actually only gain 2 ATP because you produce 2 ATP, but you have to you produce 4, but you use 2. And so we'll talk about it here in a little bit. NAD becomes NADH, and actually that's 2 of those. So this whole process of glycolysis. So here's taking a glucose, 6 carbon, 2 ATP are used up to form ADP. 4 ADP become 4 ATP, however we used to. So 4 minus 2 gives you 2 ATP. And we got some 2 NADH, or these carrier molecules. What we're going to produce, we're going to split this glucose into 3 carbon molecules that are supposed to be C's, called pyruvic acid. All right, so we got pyruvic acid. If oxygen is present, oops, what color do we want? If oxygen is present, this will occur. Okay, so if oxygen, O2 is here. We got oxygen, check. We go in to finish aerobic respiration. However, we get to a point where I've got my 2, 3 carbons, carbon, 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 carbon. We've got our pyruvic acid. All right, if there is no oxygen, we call this process fermentation. Fermentation is anaerobic. And so what will happen is, depending on the organism, and again, this is something more we'll have to talk about in class, but there's no oxygen, no O2, we've got pyruvic acid, fermentation has to occur, and depending on the organisms you can produce lactic acid, such as like in us, or there's alcoholic respiration, which again, we'll have to talk more about these, but the whole point of these is to get the NAD back, because the NAD back, well, actually right here, but really, really sloppy. I am no Bozo biology. I never will be, but that kind of gives you the idea. So that was kind of sloppy, but I think you get the idea. However, let's go back to if there's oxygen present. Let me go back to this, my mess here that I've made. So we have oxygen. So we're here. There's our carbon, 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 carbon. Coenzyme A, just what it sounds. It's an enzyme. It's important. What the coenzyme A does, it'll come in the pyruvic acid, help with the production of CO2. We've got our NAD plus. Actually, I keep putting those pluses. It'll come in, NADH. But what we're left with now is carbon and a carbon and coenzyme A. So these are just intermediaries. And what will happen is that coenzyme A, we now have acetyl-CoA. I'm going to spell this right. Again, don't get caught up in these words. They're just intermediaries. I'm more worried about the process, but no, so don't get freaking out because there's a lot of terms here, and I think sometimes teachers emphasize these intermediaries a little too much. So we got acetyl-CoA. Now we're going to go into the Krebs cycle, or also known as the citric acid. I'm looking off my sheet here to make sure. This is all in the matrix. Okay, switch up colors here. We'll go with blue. Now, we now have we've got citric acid. So we got a carbon, 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 carbon. All right, so we've got 2, 4, we've got 6 carbons. So this is citric acid. All right, it's going to go through a process. We have ADP plus, as it goes through the Krebs cycle, will become ATP. A little energy there. All right, I'm going to have to clear this so that you can see it. So if you want to pause here and get this written down. So let me go back to my pen. So again, here was our ADP becoming, that's supposed to be a D, becoming ATP. Go through, we've got some carbon dioxide again. Here, giving off, we've got NAD plus becoming NADH. Boom, we've got some FAD becoming FADH2. And then we're left with 4 carbons, and it'll just start again. So this is a cycle. It has to go around several times, actually. But what I want you to take from this, again, I don't want you to memorize all these steps, but FADH2 is produced, NADH, some CO2, and some ATP. That's the important part of this because I probably should, these two things, FADH2 and NADH, these are what are used actually to produce a lot of the ATP during electron transport. These are key. These are those carrier molecules. They drop off the hydrogens. If we go back up to the top, when I labeled the carrier molecules here, we have NADH here, FADH2. They're dropping off their hydrogens, H plus, H plus, H plus. These become NAD and FAD. They're producing the most ATP. And that's the whole point. That's the most important part of those. So, I probably confused you thoroughly, but that's fine. But what I want you to get from this, very good, different color than white. Let's go with blue. One glucose molecule will produce 6 carbon dioxides, 2 ATP, 8 NADH, and 2 FADH2 here in this process here. All right. If we go back up top, go here. One glucose will give you about 32 ATP here. This electron transport chain produces the most glucose. That was ugly, but we'll do it in class. I'll post a completed picture somewhere else. Again, this is courtesy of Kelly Rydell, but that was, I'm only at 15 minutes, so hopefully that helps. It was dirty and it was ugly, but it should work, so good luck. Pause.